species-specific recognition in sea urchins. Once the sea urchin sperm has penetrated the egg jelly, the acrosomal process of the sperm contracts the vitelline envelope of the egg. A major species-specific recognition step occurs at this point. The acrosomal protein mediating this recognition is called bindin. In 1977, some scientists isolated this non-soluble 30,500 Dalton protein from the acrosome of Strioglycendrotis purpuratus. This protein is capable of binding to degellied eggs from this species of Strioglycendrotis purpuratus. Then further, its interaction with eggs is relatively species specific. Bindin, isolated from the acrosome of this species, agglutinates its own degellied eggs, but not across those of Arabacea punctulata. Using immunological techniques, some other scientists demonstrated that bindin is isolated specifically on the acrosomal process exactly where it should be for sperm egg recognition. Biochemical studies have shown that the bindins of closely related sea urchin species are indeed different. This finding implies the existence of species-specific bindin receptors on the vitelline envelope. Such receptors were also suggested by the experiments of Wagner and Payne, who saturated sea urchin eggs with sperm. So, sperm binding does not occur over the entire egg surface, even at saturating members of sperm. There appear to be room on the ovum for more sperm heads, implying a limiting number of sperm binding sites. A large glycoprotein complex from the vitelline envelopes of sea urchin eggs has been isolated and shown to bind radioactively labeled bindin in a species-specific manner. This glycoprotein is also able to compete with eggs for the sperm of the same species. That is, if this species sperm and mixed with the bindin receptor from another one vitelline envelopes, the sperm bind to it and will not fertilize the eggs. The isolated bindin receptor from this species, however, does not interfere with the fertilization of other related sea urchins. This bindin receptor is a transmembrane glycoprotein of nearly 1300 amino acids. The bindin binding region of this protein extends into the extracellular space and probably becomes a competent of the vitelline envelope. These bindin receptors are aggregated into complexes, and hundreds of these complexes are probably needed to tether the sperm to the egg. The species-specific recognition of sea urchin gametes occur at the levels of sperm attraction, sperm activation, and sperm adhesion to the egg surface. And gamete binding and recognition in mammals. The sperm binding protein of the mouse, Zona pellucida. The zona pellucida in mammals plays a role analogous to that of vitelline envelope in invertebrates. This glycoprotein matrix is synthesized and secreted by the growing oocyte, and it plays two major roles during fertilization. It binds the sperm and it initiates the acrosome reaction after the sperm is bound. So the binding of sperm to the zona is relatively but not absolutely species specific. Species specificity should not be a major problem when fertilization occurs internally. And the binding of mouse sperm to the mouse zona can be inhibited by first incubating the sperm with zona glycoproteins. Some scientists have isolated from the mouse zona pellucida and 83 kilodalton glycoprotein ZP3 that is the active competitor in this inhibition assay. The other two zona glycoproteins, SP1 and SP2, failed to compete for sperm binding. Moreover, radio-labeled as ZP3 bound to the heads of mouse sperm with intact acrosomes. This ZP3 is the specific glycoprotein in the zona pellucida to which the mouse sperm bind. ZP3 also initiates the acrosome reaction after sperm have bound to it. The mouse sperm can thereby concentrate its proteolytic enzymes directly at the point of attachment at the zona pellucida. The molecular mechanism by which the zona pellucida and the mammalian sperm recognize each other is presently being studied. The current hypothesis of mammalian gamete binding postulates a set of proteins on the sperm capable of recognizing specific carbohydrate regions on the egg zona ZP3. Sperm zona adhesion proteins. Mouse sperm do not bore into the zona. Rather, the sperm approach parallel to the plane of the zona surface and are actively tethered there. How is the zona able to bind and retain these wiggling sperm? It appears that ZP3 can bind to at least 3 adhesive proteins on the sperm cell membrane, and thousands of these sites might be needed to keep these two cells from coming apart. There is a significant controversy concerning whether all 3 proteins on the sperm are needed to bind to the zona and what their respective functions might be. It appears that each of these proteins may play specific but somewhat overlapping roles in sperm egg adhesion and the acrosome reaction. These 3 proteins are the galactose binding protein, the galactosyltransferase, and the zona receptor kinase. The 56 kDa galactose binding protein, one critical zona binding protein of the sperm, appears to be a protein that specifically binds to the galactose residue of ZP3. So Blaine and Wasserman have shown that one of the critical carbohydrates of the ZP3 glycoprotein is a terminal galactose group. If this terminal galactose is removed or chemically modified, sperm binding activity is lost. These researchers later isolated this protein by binding ZP3 covalently to beads and passing the proteins isolated from mouse sperm membranes over them in a column. Most of the proteins passed through the column, but one, a 56 kilodalton peptide, bound to the ZP3 coated beads. It did not bind to ZP2 coated beads in a similar experiment. This protein was found to be exposed in the sperm membrane and it bound to galactose residues, strongly suggesting that it is a sperm receptor binding to terminal galactose moiety on the ZP3 glycoprotein. So purified sp56 binds to zona pellucida of unfertilized but not fertilized eggs and blocks sperm egg binding. Then galactosyltransferase. The second sperm protein that appears to be important for sperm zona binding is the sperm cell membrane glycotransferase enzyme. Shell's laboratory has shown that this receptor for the zona is an enzyme that recognizes the sugar N-acetyl glucosamine on ZP3. This enzyme N-acetyl glucosamine galactosyl transferase is embedded in the sperm plasma membrane directly above the acrosome with its active site pointed outward. The enzymatic function of this 60 kilodalton enzyme would be to catalyze the addition of a galactose sugar onto a carbohydrate chain terminating with N-acetyl glutamine sugar. However, there are no UDP galactose residue in the female reproductive tract. Although the enzyme can bind to the N-acetyl glucosamine residues of the zona pellucid proteins exactly as any enzyme would bind to a substrate, it cannot catalyze the reaction because the second reactant is missing. Therefore, the enzymes in sperm remain bound to their substrate on the zona. Then if this hypothesis were correct, we would expect that sperm egg binding could be inhibited either by inhibiting the enzyme or by adding the second reactant UDP galactose. This is exactly what Shah and co-workers found to be the case. Sperm zona binding was blocked by addition of UDP galactose, removal of the N-acetyl glucosamine residues from ZP3, addition of antibodies that blocked the activity of the galactosyl transferase, and placement of excess galactosyl transferase in the medium. The excess enzymes would bind to the zona and inhibit the sperm from binding. Moreover, mouse sperm cell membranes will transfer a sugar from UDP galactose specifically to ZP3. The sperm surface galactosyl transferase appears to be recognized a carbohydrate group on the ZP3 protein of the mouse zona pellucida. The aggregation of these galactosyl transferase causes the activation of G protein that may be important in initiating the acrosome reaction. Then zona receptor kinase, a third sperm protein that binds to the mouse zona pellucida, appears to be a 95 kilodalton transmembrane protein with 2 functional sites. The extracellular site specifically binds ZP3 while the intracellular site has tyrosine kinase enzymatic activity. This enzymatic activity is stimulated when the protein binds ZP3. This implies that the 95 kilodalton protein is a receptor tyrosine kinase and it may initiate the acrosome reaction by the phosphorylation of its target proteins. Human sperm have a similar problem, and human ZP3 stimulates kinase activity. Moreover, synthetic peptides that mimic the extracellular binding ZP3 binding domain of this protein inhibit sperm binding to the human zona, suggesting possible use as a contraceptive. Which one? ZP3. Okay. Synthetic peptides that mimic the extracellular ZP3 binding domain of this protein inhibit sperm binding to human zona. Then introduction of the mammalian acrosome reaction by ZP3. Once the capacitated sperm has bound to the zona pellucida, how does the mammalian acrosome reaction take place? The reaction is induced by the protein portion of ZP3, and this work by cross-linking the receptors for it on the sperm membrane. This cross-linking opens the calcium ion channels to increase the contraception of calcium in the sperm. Concentration. The mechanism by which ZP3 induces the opening of the calcium channels and the subsequent exocytosis of the acrosome remains controversial. So, a third sperm protein that binds the mouse zona pellucida appears to be a 95 kilodalton transmembrane protein with two functional sites. The extracellular site specifically binds ZP3, while the intracellular site has a tyrosine kinase enzymatic activity. This enzymatic activity is stimulated when the protein binds ZP3. This implies that the 95 kilodalton protein is a receptor tyrosine kinase and it may initiate the acrosome reaction by the phosphorylation of its target proteins. Human sperm have a similar protein and human ZP3 stimulates kinase activity. Moreover, synthetic peptides that mimic the extracellular binding domain of this protein inhibit sperm binding to the human zona, suggesting possible uses as a contraceptive. Then secondary binding of sperm to zona pellucida. During the acrosome reaction, the anterior portion of the sperm plasma membrane is shed from the sperm. This is where the ZP3 binding proteins are located, and yet the sperm must still remain bound to the zona in order to lyse a path through it. In mice, it appears that the secondary binding to the zona is accomplished by proteins in the interacrosomal membrane that bind specifically to ZP2. Whereas acrosome intact sperm will not bind to the ZP2 glycoprotein, acrosome reacted sperm will. Moreover, antibodies against the ZP3 protein will not prevent the binding of acrosome intact sperm to the zona, but will inhibit the attachment of acrosome reacted sperm. The structure of the zona consists of repeating units of ZP3 and ZP2, occasionally cross-linked by ZP1. It appears that the acrosome reacted sperm transfer their binding from ZP3 to the adjacent ZP2 molecules. After a mouse sperm has entered the cortical granules, release their contents. One of the proteins released by these granules is a protease that specifically alters ZP2. This inhibits other acrosome reacted sperm from moving further towards the egg. It is not known which one of the mouse sperm proteins bind to ZP2. In porcine sperm, secondary zona binding appears to be mediated by proacrosin. Proacrosin becomes the protease acrosin that has long been known to be involved in digesting the zona pellucida. However, proacrosin is also a fucose binding protein that maintains the connection between acrosome repeated sperm and the zona pellucida. It is possible that proacrosin binds to the zona and is then converted into the active enzyme that locally digests the zona pellucida. So in guinea pigs, secondary binding to the zona is thought to be mediated by the protein PH20. Moreover, when its inner acrosomal membrane protein was injected into male or female guinea pigs, 100% of them became sterile for several months. The blood sera of these sterile guinea pigs had extremely high concentrations of antibodies to PH20. The antiserum from guinea pigs sterilized by injections of PH20 not only bound specifically to this protein but also blocked sperm zona adhesion in vitro. The contraceptive effect lasted several months, after which fertility was restored. These animals had been temporarily sterilized by these antibodies. The human analog of the PH20 protein is not yet known, but certain sperm antigens display a similar pattern of localization in the sperm. Similarly, the human zona proteins and their functions are not as clearly established as those in mouse. And nevertheless, this experiment show that the principle of immunological contraception is well founded.